All right, sorry about that. So today is right there, the 13th for odd classes. If you're watching the video, then it was Monday. So not next class, but the class after that is our quiz. So if you're an even day student watching this, that was Monday, that was Wednesday, that was Friday. So this Friday, even classes are taking our quiz. And then if you're in the odd day classes, 3rd and 5th, then yours is after the weekend. Remember, we have a 3-day weekend coming up. Next week, we are off on Monday in observance of Martin Luther King Day. I think his actual birthday is the 20th, but they always switch it to where it's a Monday. Kind of like Thanksgiving is always a Thursday. So when you come back from the 3-day weekend, we've got our quiz. As far as what's fair game on the quiz and what the style is, it's 10 questions and DNA structure. A's go with T's, G's go with C's, why's that, anti-parallel, all those kinds of things. DNA structure matters, replication. We did that right before we left for break pretty much. That was like unzipping DNA with helicase. I have primase put down primers. I have DNA polymerase 3 come in and put down new bases, that kind of thing. Replication. Transcription, we're doing that today. And then RNA processing, we're doing that today. It's possible that we'll finish transcription and RNA processing today. What's likely is we'll have a little bit of RNA processing left over that we learn next time. So today we'll be doing transcription and RNA processing. So you'll be almost ready for the quiz, content-wise, maybe not studying-wise, but you'll be almost good to go after today. We'll probably have a little bit of that left over. And then our test for even days is the 27th. For you guys, odd days, it's the 28th. So, yeah, and then our next topic after this is we combine evolution and ecology. So it's kind of different. Instead of being on all this small molecular stuff, it's kind of more like big ideas like, you know, populations and stuff like that, predators and prey and all that. So any question about our schedule up here? So not next class, but the class after is our quiz. Cool? If you are following along in your notes, then you would be at, thank you. You would be at this page. I don't have it in front of me. Oh, there we go. So if you're wanting to follow along in your notes, then I would literally just take this physical page of paper, and I want to be on the back of it where it says AP Biology, Protein Synthesis, Transcription, and Translation. It says Protein Synthesis, Transcription, and Translation right here. Right there. Flip it over, it'll say Protein Synthesis, Transcription, and Translation. Huh? Yeah. Thank you. It should be. You'll probably have to go about 3 or 4 deep because lab is on top. Yeah, I think we have 3 lab things on top, so it'll probably be the 4th layer down. Protein synthesis, transcription, and translation. Thank you. All right. So I'm pretty sure I've shown you guys this picture in the past, haven't I? Does that look familiar or not? Okay. And it depends on the class. So if you switched in here, you used to be in my 5th period or 6th, and now you're in here, you might be like, hey, they said they've seen that, and I haven't. Sometimes you guys will ask questions in class, as this has happened in all of them, and so I'll go over and I'll be like, hang on just a second, I got a picture that relates. So what was it that was strange about this guy? Yeah, that's supposed to be thigh muscle, but he has like bone tissue growing in his thigh muscle. And this is a bone density scan. So a bone density scan works a little bit differently than an X-ray. So if you're like, wait a second, I thought if that's an X-ray that the background would be dark and then that would be really, really bright. It doesn't work exactly like that. It's not completely opposite, but it's kind of the opposite. Uh-huh.
Like the integral parts of like each bone supposed to be that like light colored? Like I mean.
In a bone density scan, yes. So for here, it's weird. Cartilage is showing up as darker, so that's what seems strange. But otherwise, you might be like, hey, I would have thought your bone should be really dense right there. Like that's almost non-existent. Like does this person have like severe osteoporosis or something like that? So to my knowledge, this is normal except for that. All right, so then we do another scan of the same guy, and if we look at it, we notice that like that's his right thigh, that's your knee. So your knee's kind of weird. Your knee bone and like my knee right here is, you don't have a knee bone, but your knee bones. Your femur, your thigh, sits on top of your tibia. We often call your tibia your shin. Your fibula is back there in the muscle, but like you can't really feel your fibula. But your tibia is like the bone in the front, and then this is your femur, and they just sit on top of each other. So what is the thing that's like actually attaching? Like if I run and I cut, my femur doesn't like fly off, my leg doesn't break in half. So what's keeping them attached? Yeah, ligaments. So tendons attach muscle to bone, and then ligaments attach bone to bone. So the most common one that you hear about is the ACL. So I have the anterior cruciate ligament. It's more on the front. It's more on the anterior side. Then I'll have like the MCL, the medial. So they have these names that sort of tell you where they're located. But imagine I just have like really, really strong bungee cords or really, really strong rubber bands that are just like, I drill in a bungee cord into this bone, my tibia, and I drill in a bungee cord into that one, and like that's what's holding it together. So I've got a few in the middle that are like holding it together, and then I have a few on the side. And of course, if my thigh muscle is pretty strong and it's kind of pulling there, it kind of like stabilizes it too. But really, all that's actually holding them together are like those ligaments. And so that's why athletes, if I'm running straight, I'm not very likely, unless I just like suck, I'm probably not going to like tear my ACL. But if I like run, stop really hard and twist, then now I am much more likely. Let's see. Oh, thank you. But if I run and stop really hard and twist, then now I'm much more likely to, do you like tear your ACL by just running straight?
No, mine was contact.
Okay. So if you're just running straight, you're not likely to tear it. But if you run, stop really hard, like plant and twist, something that you might see in basketball more or football or something like that, that's when you're putting just tons of pressure on there, because now I have this like this little rope, this tendon in there, and I'm going to take it, push on it, twist it, and turn it like that. And so whenever I twist, if my tibia is planted because I'm like wearing cleats, and my femur twists and my tibia doesn't, that's how I end up like tearing my ACL. So some of us anatomically we're put together differently, and so we tear ACLs more easily than other people. Yeah.
I was going to ask, why is it that girls?
So the way that girls' knees are often built is slightly different. Mostly it's size. So I've heard multiple times that females are more likely to tear it. I'm guessing males tend to have more muscle mass. I'm not saying females can't be just as strong as males. I'm saying that if this person is an average athlete and they work out an average amount and they have average motivation, the male will be stronger. Not as much on lower body. Males will be stronger on lower body. Males, you notice the biggest difference in upper body. So I would guess it's the musculature around it that's stabilizing it. If I'm a football player, I have a friend, Darren, he played football in high school and college, and he was a wide receiver. And he tore his ACL and kept playing. But his legs were huge, and basically just like, hey, as I'm running, as long as I flex my legs hard enough, my leg muscles are holding my knee together. So I bet it's muscle mass, but I don't know that for a fact. But yeah, I don't know what the ratio is, but females tear ACLs a lot more often than males. If you're comparing the same sport, basketball to basketball. Football, it's different. You know, but if you compare baseball to softball, basketball to basketball, like equal sports. Yeah. Other questions so far? Cool. All this stuff is super fascinating to me. So with this guy, there are different versions of this disease. One is called MOP, one is called FOP, but essentially your muscles are slowly converting into bone. So whenever I injure a muscle, you or me, if I damage a muscle and I destroy 10,000 muscle cells, I go in and I turn on the muscle genes and I make 10,000 new muscle cells to replace it. Just like if I lose 50,000 skin cells, then I replace it with 50,000 skin cells. However, if I have MOP or FOP, then I accidentally turn on the bone genes. And so whenever I'm repairing muscle, I'm repairing it with bone. And so you might not know a whole lot about anatomy, but you know that ain't right. You know? So we're going to kind of look at how that happens. So here's my cell. And this one for transcription and translation, I pretty much do explain it all with one big picture, so a little bit like cellular respiration. If you're the type of person that draws along with me, I'll have this picture. Next class when you come in, I'll add on to this picture and so forth. So if you just took like a whole page, a blank paper in your spiral or whatever, that's kind of how often we'll use this picture. So I got a nucleus right here. My nucleus is going to be pretty big because I've got a lot of stuff that happens inside the nucleus. A lot of stuff is going to happen inside the nucleus. All right. And then I'm going to probably have like 2 colors going. I feel like I'm Bob Ross. We get a little bit of phthalo blue, cad yellow. All right, so I got some DNA in there. I'll use red and blue. That's a letter P. That one's a letter R. And that one is a T. So P, R, and T. Leave a little gap right here. P, R, T. Kind of like that. So if these can be one color and that can be a different color, it'll be a little bit easier to kind of follow along. If you've only got a pencil, then it doesn't matter too much. The one that's red up here for me is a muscle gene. This one down here, it codes for bone, so that just says bone. So this is like a skeletal system thing, and this is like a muscular system thing. So I got these 2 genes. I got the red one and the blue one. All right, cool. So P and R and T. While you guys are doing that, I'm going to grab a file real quick that might help us out. So there's a P, an R, and a T. All right. So back in the day, in the 1990s, they started it, and they finished a rough draft copy around 2003. They did the Human Genome Project. We've mentioned genomes a little bit here and there, and they basically took one cell from one guy, popped open the nucleus, and just sequenced all 23 of his chromosomes. And they're like, this guy's alive. He's pretty old, so this is normal enough to at least make it to age however old he was, 60, 70, 80. So we're just going to call this like, this is normal. Just like you had some other human, you got some guy that lives a pretty long life and he doesn't have any known major genetic diseases. You're like, all right, well, this must be like normal enough to live. So they sequence his DNA. And so along in here, you now have to just figure out like, what is what? So I download a little bit of the human genome, and this is it. Okay, now, students will always wonder, how many pages is it? This is 57 pages, but that's just a small chunk of a spot within a band on chromosome 2. So you might be like, all right, well, what about like the entire human genome? Like, how many pages is that? Or like how long is that? So I got you a little video clip here. Oh. Get a little bit more volume. Okay, here we go. So if, like for example, let's say that I wanted to print one of those pages. So I've got the whole human genome downloaded, and I wanted to print page 25. But instead of hitting print page 25, I accidentally like hit print all. I don't know if you've ever done this. You got a file that's kind of long. You just wanted to print this page or that page, and you hit print all. Bless you. Then what would happen? So at the font that I was using, at the spacing that I was using, this is what happens.
Think of how many letters fit on a single page. And now imagine a stack of pages 300 feet tall. That's how much information is stored in the DNA inside every human cell, the entire human genome.
Now, whenever they print it, they only print one side of the DNA. So if you were going back and looking at mine right there, if you're trying to get like that and that to match or that and that, they don't. They only read one side. If I know that this side is G, 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 I don't need to read this side. I know that it's supposed to be C, 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 right? Assuming no mutations. So they're just reading down one side of the DNA. So that's what this is. This is one side. And then they just number the bases. So if I'm looking at this one, for example, then what I'll see is like right here. That means this is base number 37,561. That's 37,562. All the way to right here, this is 37,620. That's base number 37,621. So they just group them in tens. So that's what those numbers are. And turns out the entire human genome is around 3.1 billion base pairs. And you guys have gotten so used to hearing the word billion that you don't realize how big of a number it is. If I print off 3.1 billion letters, this size font, this spacing on single-sided paper, it's 300 feet tall. So that's a 30-story building, right? 300 feet, that's a 30-story building, and Timber Creek is a 2-story building. So that's pretty tall. Anyway, so what we find is inside the DNA, a lot of it, we don't really know what it does. In fact, most of it, we don't know what it does.
If you sort through the 3 billion letters that make up the human genome, you find some surprising things. Only about 1% of the 3 billion letters directly codes for proteins. Of the rest, about 25% make up genes and their regulatory elements. The function of the remaining letters is still unclear. Some of it may be redundant information left over from our evolutionary past.
So they finished the Human Genome Project officially a couple of years ago, Anytime you have big, long repeats, those are hard to sequence. So like telomeres, tag, 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 tag. In 2003, they didn't have all those sequenced. They came up with a different way of reading DNA, and now the repeats were like readable. So if you're like, hey, they finished the rough draft in 2003, and then they finally finished the real thing in like 2020 something, what took so long? They were like 98 point something % done back then. Why did that last percent and a half take so long? That's why. Anytime that you just have these really, really long repeats, it was harder to identify them. So what we found is, she said 1%. We think it's about 1.5 now. This video is slightly old. So about 1.5 maybe codes for protein. That means that like codes for a protein, an enzyme that does something. So the way that I teach this to like say my Bio 1 students is I'll be like, here's DNA, we convert it into RNA, and then that RNA codes for a protein, and then that protein helps build your heart valves. And so maybe there's 500 genes that help me build normal heart valves. And if all of them are correct, I have normal heart valves. Some people though, gene number 212 and gene number 115 are messed up, and that's why their heart valves are messed up, and that's why they have a disease called this. These genes over here, these code for building a normal kidney. If you got them all, great. If this gene is screwed up, that causes this problem, and you have this disease. If this other gene that codes for kidneys is screwed up, you have this other problem, and you have this other disease. So this is how diseases happen. But only about 1.5% of our DNA actually codes for it. We now think it's maybe more like 30%. She said 25, but about 30% is just turning genes on and turning genes off. It's like administration. They aren't doing anything. They're just bossing around the workers. So you can kind of think of it like this. Proteins, admin, or you can think of proteins as like the workers. They actually do stuff. And then these guys are just bosses. They don't actually do anything. They just make sure the workers are doing their job. So a principal, an assistant principal. An assistant principal is supposed to help me do my job. But if every teacher is perfect, then you kind of don't need that person. If every student is perfect, you don't really need them for anything. Just like managing a restaurant. The manager isn't serving food, they aren't cooking food, or anything like that. If everything's perfect, if the restaurant's working perfectly, the manager is just literally sitting there with nothing to do. You know what I'm saying? So if I'm the CEO of Chick-fil-A, I haven't fried chicken in years. I just tell people to do crap, and I fire people, and I hire people, and I collect checks. So most of your DNA, we don't know what it does. So 2/3 of your DNA, it's like, what does that do? And we're like, we don't know. We'll go into your DNA and remove chunks. We'll take a mouse, and we'll be like, hey, what happens if we just delete this whole page of DNA, splice it back together, see if we can grow this mouse up? So we go in, we fertilize an egg and all that, and then we like cut out some DNA, and then we say go, and the mouse grows up just fine. So we're like, I don't know what that does. It's almost like I'm playing my favorite video game, and I went in there and I just removed like 1% of the code, and the game still seems to play totally fine. You're like, I don't even know what that does. When it's missing, it seems to make no difference. Sometimes we remove a chunk, and all of a sudden it doesn't work, but we still don't know what it actually does. So 2/3 about of our DNA, we still have no idea what it even does. About 1.5% are the workers. Kind of interesting. So what we're going to focus on are these two parts. Gene regulation is like the administrator, the bosses, and then the proteins are like what actually do stuff. Okay, so over here we have a P, a R, and a T. So those all, of course, stand for something. So a letter P is a promoter. That's a sequence of DNA that functions as a promoter. It's still just made of A's and T's and G's and C's, but it is a promoter sequence. A promoter sequence. Eventually, I'll be kind of using this part of the board for like a future drawing, so I'm just kind of putting this somewhere out of the way a little bit. A promoter sequence. So that right there is a promoter. All right, cool. But it's just still a set of A's, T's, and G's that stands for I'm a promoter. I'll tell you what a promoter is in just a second. R is a regulatory sequence. So it is a regulatory sequence. Again, it's still just a sequence of A's and T's and G's and C's, but its job is to regulate. So a promoter sequence, a regulatory sequence, and then the last one, the T, is a termination sequence. Termination sequence. A termination sequence. All right, so same thing down here. Promoter, regulatory, termination sequences. Now, if you had to guess, what do you think a promoter is? This isn't easy for sure. It's not that. Once I tell you one of them, you'll be able to get one of the other ones. What do you think a promoter sequence represents? Yeah, it's kind of the beginning. It's the beginning of a gene. So I think of it like this. If I've got a sentence, I'm going to make up a language right now on the spot. This is my own pretend language. All right. You speak English, so I can do anything I want in my language, but if you had to guess, how many complete thoughts are up here in this pretend language? 3. Why did every single person guess 3? What's your logic? Yeah, punctuation and a capital. Sentences usually end in punctuation. Sentences usually begin with a capital letter. Things can throw us off. If halfway in here I had like Dr. or Mr. or Mrs. or something like that, I'd have a capital and a period, so that could mess with me a little bit. But in general, it looks like that. Do you know what this actually says? No. So you don't know what it says, but you know that's probably the beginning. That's probably the end. That's probably the beginning. That's probably the end. I have no idea what it says. There are certain sequences that are common at the beginning of genes, and we call those promoters. So once I kind of learn, I can just feed this into a computer, and I can be like, hey, here's something that we know is a promoter sequence. It's T, T, G, G, C, G, G, C, C. They're usually rich in C's and G's. G, C, C, T, A, whatever. Here's another promoter. Here's another. Here's another. And I just feed it to a computer. And then I search, and I'm like, find things that look like a promoter. So here's the human genome. I have no idea what any of it does. My computer hits on this chunk right here. And my computer's like, hey, we're like 80% confident this is a promoter. And I'm like, all right, cool. This is probably the beginning of a gene. Then I scroll on down, and my computer hits somewhere down here. And it's like, hey, we're like 75% confident this is a termination sequence. A termination sequence, if a promoter is the beginning of a gene, then terminate means to end. So a termination sequence is the end of a gene. So we might be like, hey, I think this is the beginning of a gene. I think this right here is the end of a gene. So I think this is a gene that codes for something. What's it code for? I have no freaking clue, but I'm pretty sure that's a gene. We might discover a brand new animal or a brand new plant. Look at it for a little bit and be like, hey, we think that this animal has 5,000 genes. And you're like, oh, what do the 5,000 genes do? You're like, we don't know. We don't know what any of them does yet. And you're like, well, how do you know you have 5,000 of them? Well, we kind of found about 5,000 things that look like this. Just like if I'm looking at a Spanish or a French text, I can be like, I think you have about 10,000 complete thoughts here. Oh, can you read French? No, but like, that looks like a sentence, that looks like a sentence, that looks like a sentence. And they're like, oui. So I'll find certain sequences that are promoters, certain sequences that are termination sequences. Then what I'll do is I want to figure out what that thing does. So the way that I do it is this. I make it glow. So we have genes that are found in nature in animals that glow in the dark. So some of you guys have maybe seen like glowing jellyfish at like a, I don't know, some sort of zoo or on television, some sort of David Attenborough thing or something like that. So jellyfish can sometimes like pulse and they'll glow green, red, blue, whatever. So what we'll often do is this. We'll be like, all right, that looks like a promoter. That looks like a terminator, a termination sequence. We don't know what the gene does, so what we're going to do is we're going to go over here into this jellyfish. Here's this jellyfish, and we know for certain that this right here is the glow green gene. So we're just going to take that and like splice it in here. Right there. So now, when this gene, if it makes a protein, it will be a protein that glows green. So now I just birth this mouse, this moth, whatever it is that I'm doing this experiment on, a fruit fly, and I just see what glows. So maybe I got a moth, and it looks like this. So I'm looking at this, and I'm like, okay, I've got this moth DNA. That looks like a promoter. That looks like a termination sequence. What to do? I don't know. All right, well, make it glow. We make this moth, and now anytime I shine UV light on him, his eyes glow super bright green. So it's like, okay, well, this is not a question anymore. We've narrowed it down. It has something to do with eye development. We don't know exactly what, but it doesn't seem to be related to like his wings. His legs look normal. All these other internal organs are fine. So like, hey, that gene is now something to do with eye development. We narrowed it down. And all we did was we started by taking its genome and searching and being like, that chunk looks like a promoter, the beginning of a gene. That looks like a termination sequence, the end of a gene. This is probably a gene. Make it glow, and we'll see if we notice anything. Now, a lot of times we don't notice anything, but sometimes we do. Those are mice. And we clone them. So these are all twin brothers, twin sisters, whatever. The only difference is these guys got the glowing green gene spliced in. And apparently that glowing green gene coded, or that gene coded for skin, and so now their skin glows green. This guy is his exact identical twin with that one exception. So in science labs, we just clone everything, change one gene, and sort of see what's different. Makes sense. If I'm trying to figure out how a car works, I could be like, all right, my car's running. Just going to disconnect that wire and see what seems to be different. Okay, put that on. Now it's running. I'm going to disconnect this thing and see what runs different. And eventually I can figure out like what this thing does, you know? So when I disconnect this, the brakes don't work. All right, that has something to do with the brake system. I can investigate further. So a promoter sequence is the beginning of a gene, and it is where RNA polymerase lands. It's the beginning of a gene, so you could say it's the beginning of a gene. And because of that, that's where RNA polymerase lands. We learned about DNA polymerase earlier and DNA replication, but when we want to make RNA, we use RNA polymerase. So this one is RNA polymerase, known as RNAP on the street. It's beyond AP biology, but this happens to be the second one we discovered, so it's technically RNA polymerase 2. That doesn't really matter much, but in case you're keeping track. If you're looking up, if you're going back and searching anything on the internet or whatever, it's going to be like, this is RNA polymerase 2. So RNA polymerase 2 lands on the promoter sequence, and that's how it's going to start reading the gene. So let's say I don't have MOP or FOP, and I'm running along, and I hit my thigh muscle, my hip muscle, on the corner of this, and I damage some muscle cells. So here's what's supposed to happen. The muscle gene turns on to make new muscle cells. And so RNA polymerase, RNAP, will land on the promoter. So it'll land here like this. that is RNAP. And it lands on the promoter. Then what it does is it just like slides along, unzips the DNA, and reads it. Before, we were just replicating DNA. When we replicate DNA, I'm just copying it. I don't have to know what it means. Now we're actually reading it and doing what it says. I can copy something, back to my analogy of French, I can copy this French book word for word perfectly and have no idea what it means. That's like DNA replication. In DNA replication, I'm copying it. I'm not reading it and doing what it says. I can copy this cookbook and not cook anything. When we're doing transcription and translation, we're actually opening it up and reading it and doing what it says. So now we're actually trying to do what it says. So RNA polymerase lands on the promoter and then it like cruises along and it'll like unzip this DNA. It's kind of odd. It doesn't need helicase. It does the unzipping itself. So it'll like land on the promoter, cruise along here, unzipping the DNA, and it's going to start building RNA from it. So my RNA polymerase, anytime it's going to land, it lands at the beginning of a gene, the promoter sequence. It shouldn't land anywhere else if everything's working fine. And it'll land, it'll cruise along, it has two jobs, it does the unzipping, and then it starts building a piece of RNA. So that red strand right there that I just added, that's RNA. So I got a muscle gene, the beginning of it's the promoter. The end of it is the termination sequence. RNA polymerase lands on the promoter, cruises along, and how does RNA polymerase know when to quit? What tells it to stop? Yeah, termination. So my termination sequence binds a terminator protein. Or you could say terminator protein lands there, however you want to say it. It binds a terminator protein, and that blocks RNA polymerase from going any farther. So it's almost like RNA polymerase is like a train cruising on train tracks, and then I just have like a 50,000-pound boulder sitting on the train tracks. It's like, all right, this is where I stop. Like there's a boulder in the way. So a terminator protein, not always, but often, it just physically blocks me from going any farther. Like I'm cruising down these train tracks, and then there's a boulder, so this is where I stop. Okay, remind me really quick, what bases did we use in DNA? We used, what did A match up with in DNA? T, and then what was the other pair? C and G. And then RNA was slightly different. What was different about the bases in RNA? Yeah, I had U, uracil, in place of thymine. So in DNA, A goes with T, G goes with C. In RNA, A goes with U, G goes with C. So I'm fully aware that you learned that back in Bio 1, but if you're a senior, it's possible that was like 3 years ago. So A goes with U. The mistake that people make that are older that they kind of forgot is they're like, wait, I know that uracil was like in place of thymine, but was it like in place of thymine? Or did it join with thymine? You know what I mean? Like A's go with U's. It's not T's go with U's. So a lot of students will just be like, wait, I knew that it was like something to do with thymine. Does it join with it or does it replace it? So T's don't exist in RNA. U's do. All right. There was also a difference of the sugar. What was the name of the sugar in DNA? Deoxyribose. And then what about RNA? What's the name of the sugar, the carbohydrate in RNA? Ribose. So the sugar in DNA and RNA are very slightly different. U's and T's are very slightly different. For example, if I told you, if I put a picture of T up here, the molecular structure, and I said, look at it, here in a little while, I'm going to put another picture up there, and you're going to have to tell me if it's a T or not. If I put a picture of T up there, and then took it down, talked for 5 minutes, and then I put a picture of U up there, and I asked you what it was, you'd say T. They're almost identical. They're not, but they're super, super close. So they're very slightly different. So DNA and RNA are just very slightly different. However, remind me, why does my DNA always have to be inside the nucleus? Why can't it be outside the nucleus? If DNA is outside the nucleus, what happened? Who was it that wrecked it? DNase. So DNase will destroy DNA. Does RNA look very, very slightly different? Yes. So does DNA recognize it as something that it wants to destroy? No. So DNA can never leave the nucleus. Can RNA leave the nucleus? Yes. So let's say I want to read the gene for DNA and build whatever it says to build. Let's say this says build some muscle. Well, the protein that helps me build muscle, let's say, is this big. If I just read my DNA and build the protein that it says to build, the protein I build is this big. Does it look like this protein is small enough to fit through those nuclear pores? Nope, it's not. This protein, to do its job, has to be out here in the cytoplasm. So, can the DNA leave the nucleus? No, not without getting wrecked by DNA. So we'll have to do something about DNA. But this protein has to be built out here. So how do I get the message of DNA out here in the cytoplasm to build the protein? I convert it into RNA. So that's why we convert DNA into RNA. RNA can safely leave. Why? It's very slightly chemically different. It has U's. It has ribose instead of deoxyribose, so DNase leaves it alone. So we want to convert DNA language into RNA language. All right, so I get this piece of RNA, and then don't draw this part yet, because I'm going to show you what you learned in Bio 1. So then what you learned in Bio 1 is we just did transcription. Your teacher will have something like this. Or if I was your teacher, they'll have like pretty much exactly this. I'll be like, transcription happens in the nucleus, and like DNA is converted into RNA. And so if your teacher's drawing a picture, they'll be like, we unzip DNA, we make RNA. And then the RNA leaks out of the nucleus. And even us, earlier in the year, in Unit 2, who was it that landed on that RNA to read it and build protein? What organelle reads the RNA to build protein? Yeah, ribosomes. So then we'd be like, ribosomes land on there and read it. And that was kind of the end of transcription. Okay, we're going to add on to that. So we're going to back it up a little bit. Okay, now, P, the promoter, that was the beginning of a gene. That's where RNA polymerase lands to build this strand of RNA. It knows to stop when it gets to the termination sequence. It'll be a terminator protein. Regulatory sequence. A regulatory sequence can bond a repressor or an activator. Let me make that an or. Or it can bind a repressor or an activator. It can bond to a repressor or an activator. Repress or activate. So its function is it's basically an on-off switch. Obviously, if I bind a repressor protein, you're like, that sounds like you're turning this gene off. If I bind an activator protein, that sounds like we're activating the gene, like we're flipping the switch on. So the overall function of a regulatory sequence, if you want to think of it this way, is it's an on-off switch. The promoter is kind of the beginning of a gene. Begin. Termination sequence, that's the end of a gene. The regulatory sequence is the on-off switch. So those are the lights. Right now, the switch says we're in the off position. If I flip the regulatory site, now I express the lights. They do their job. They're in the on position. I started chugging a 2-liter of Coke every day. And so now my body's like, all right, we need to turn on those insulin genes. We don't have enough insulin. This guy's blood sugar is crazy. Or I lift weights. And they're like, all right, if I get injured, if I'm walking along and I hit my thigh and I damage 10,000 muscle cells, I should replace it with 10,000 muscle cells. But my body treats it differently if I exert myself and I'm not strong enough. So if I lift weights and I destroy 10,000 muscle cells, my body will replace it with 10,010 muscle cells. So now I should be slightly stronger. And so then I work out again. I tear some muscles again, and this time, whenever I rebuild them, I rebuild my muscles at 101%, 102%. So every time I lift and tear down muscle, and lift and tear down muscle, I get stronger and stronger and stronger and stronger. So regular damage from an injury, I probably just 100% repair it. But damage from lifting, my body will repair it stronger. Okay, so I need to turn this gene on, and I need to turn this gene off. How do I turn that blue gene, off? I don't want to grow bone in this cell. I don't want any skeletal system in there. How could I turn it off? This one's on. I'm reading it right now. How can I turn it off? Where's the on-off switch? The regulatory sequence. Does it look like there's anything stuck to the regulatory sequence right now? No. So if that's the case, I'm going to read this gene and I'm going to read this gene. That's what's wrong with that guy that had the bone growing in his thighs. So what do I need to do to turn this gene off? Yeah, I'm going to need one of these repressors. So if I have a repressor, I can have a repressor. I'll do rep. A repressor can bind to like a regulatory site. It might just stick to the regulatory site. It might even cover the promoter a little bit. So this version's slightly easier to understand, so I'll go with that one. I've got a repressor. That repressor can bind to a regulatory site. So it's binding to a regulatory site, but doesn't it look like that repressor is kind of covering the promoter? So can this RNA polymerase currently as is, does it look like it can land on that promoter? No, because somebody's in its spot. It's blocked. So that gene is off. Kind of like if I'm a brand new student to this class, I switched from 6th to 3rd or 5th to 3rd or whatever. I walk in and I'm like, well, I'm not sitting there or there or there or there. It's like, all right, I guess I can sit back there in one of those two. Those are open. And this seat's open. So there you go. I'm going to sit right here, I guess. Same thing. RNA polymerase doesn't know where he's supposed to go. He just knows that he can't land there, and he can land there, so he lands there. So here's what's so shocking to me. This seems like this would just be full of errors. My skin cell, I know I've said this part before, but this skin cell has all 21,000 genes in it. This brain cell has the exact same 21,000 genes in it. Why is my skin cell a skin cell and my brain cell a brain cell? My skin cell, all the skin genes are on. Why aren't I growing brain in my arm right there? Because all the brain genes are off. All the heart genes are off. The lung genes are off. The only ones that are on are skin genes. And then right below it, the muscle genes are on in my muscles. All the skin genes are off. So I don't have skin growing in the middle of my arm. But the genes for brain are right here. The genes for heart and lungs are all right there. So in my thigh muscle, it should look like this. Only the muscle genes should be on. Every other gene should be repressed. In my thigh, when I find these 1,000 brain genes, all of them should be off. What happens if the brain genes are accidentally on? I will now grow a brain in my thigh. So now think about it this way. I need you to come visit Timber Creek. You've never been here. You're a guest. You're like our FBI guest. If I wanted to make sure that he parked in a specific spot, I could say, hey, there's a spot out front. It's number, you know how they have them numbered. It's spot number, faculty, so FAC 39. And he might find it and he might park there. Could he theoretically park somewhere else different? Sure. So how could I keep him from doing that? I put a boulder in every single parking spot except faculty number, whatever I said, 37. Now he is forced to park there. That's how genetics works. I'm RNA polymerase. I'm going to land and read every single open gene. So I'm going to land right there at that promoter and read that gene. I'm going to land right there and then those two spots right there. The reason I'm not going to read any of these genes is you guys are repressors and you're in my way. And it works. So in this skin cell, I have to properly turn off thousands of genes to be normal. And in the skin cell right next to it, I have to turn off thousands of genes to be normal. In my brain, I have to turn off these thousands of other genes to be normal. And we pretty much got it right. How is that even possible? Right? I have 20,000 genes, and every single one has to have the proper on-off configuration in every cell, and I'm made of tens of trillions of cells, and it pretty much works. I've never heard of somebody growing a brain in their arm. I've heard of somebody growing a bone in their thigh, but it's actually shocking that this isn't way more common, isn't it? It's bizarre. So, promoter, beginning of a gene, termination sequence, the end. If there's nothing really blocking me, then that gene is on. If there's something blocking me, then that gene is off. We'll get into activators a little bit later, but overall, does this kind of make sense? Outside of the magnitude of weight, how does this not screw up? Okay, next. The next thing we want to do is we want to process our RNA. So back in 9th grade bio, like I had up here a little bit ago, we would just say, now my RNA leaks out of the nucleus, a ribosome lands on there, starts building protein from it. That was great for Bio 1, or for Chapter 2 in AP Bio. But there's more to it. So the next part is RNA processing. RNA processing. Some books call it RNA editing. That's fine. Processing, editing, whatever. We tweak our RNA. The AP Bio test loves RNA processing. They love a lot of questions to where it's sort of like, hey, unless you had a pretty intense Bio 1 teacher, you probably didn't learn this in Bio 1. You would have to learn this in like AP Bio. So they'll ask you easy questions. What does A go with? What does T go with? But then they might ask you like why. And most people don't learn that in Bio 1. So RNA processing, there are 3 things that happen. The first thing is called a 5' cap. So we add a 5' cap. The second thing that happens, and of course, here in a little bit, I'll like tell you what the heck that means because I know that doesn't really make much sense, but I'm going to add a 5' cap. The second thing that happens is splicing. I don't know why, but such a satisfying word, splicing. It's probably just me. Splicing, we'll find, becomes really interesting. Once we get into the details of splicing, it's actually something that if you find this class interesting at all, you'll be like, that's pretty cool. Splicing. And then number 3, leave a pretty good gap for splicing. Number 3, poly A tail is added. So add a poly A tail. The word poly, P-O-L-Y, just the letter A, and then tail. A poly A tail. These are the 3 things we do in RNA processing, or we might call it, like I said, RNA editing. All right, so 5' cap is added. What that means is normally my RNA is like put together a certain way. You know how there's like 3' and 5' and all that crap from like before we left for winter break? A 5' cap is just on the front. I think of cap as the top. I don't know what your all's grandpas are like, but my grandpa, that would be the equivalent of your great-grandpa. But my grandpa, he didn't call like a baseball hat a hat, he called it a cap. You guys ever heard of your hat being called a cap? So to me, that makes sense that that's on the front. And then obviously a poly A tail, like that's at the back, that's the end. So I've got this piece of RNA, I put a cap on the front of it, and if I'm adding a tail, that would be like on the end of it. So the job of a 5' cap is this. What it is it's just a nucleotide. Normally, if I'm adding like A's and U's and G's and C's, I'm adding them like this. It's essentially this like modified upside down guanine. You don't have to know that, but just realize it's like this modified base. It happens to be like upside down. So instead of joining like this, it joins in this weird configuration like that, and that means this is the start of RNA. It's kind of the equivalent of putting a capital letter. That means this is probably the start of a sentence. So we take this base, and it's just weird, and we call it a 5' cap. So it's just a regular nucleotide. If it was like this, everything's cool, but instead it like joins in this odd way. So that's a 5' cap. That means this is the very front of my piece of RNA. So my RNA will look like this. I just kind of designate it as a box with a 5', like that. Those are the bases sticking up, A's, U's, G's, and C's. So if I add a 5' cap, it's just a modified base, but usually we designate it something like that. Books will designate it as kind of like a square or a rectangle with like a 5'. The next part, splicing, is super cool. So if I look at my RNA, it turns out I don't use it all. I only use parts of it. I cut out parts. So now if I look at a more detailed version of my RNA, if I kind of color code it, I've got red pieces and blue pieces. The color doesn't matter. If you don't have red and blue, that's fine. If you can somehow, like if all you have is a pencil, then make this like really dark, thin and light, really dark and heavy, thin and light, really dark and heavy, thin and light, something like that. So here's what's kind of weird, and we won't get all the way through splicing. But here's what's strange. These are wiki sticks. You guys ever seen wiki sticks? Okay. There's a Mexican restaurant on 377 called Fresco's. Have you been there? Some of you guys gasped. They're like, oh, I do remember wiki sticks. Okay, so there's this Mexican restaurant. I don't know if it's still there. When you drive by, ever since COVID, it's very difficult to even tell, like, is this place open or not? But it used to be really busy. And what they would do is, let's say you bring in your little kid. So when my son was little, we'd go to Fresco's. And instead of giving you like a color page, if you go to Chili's or something, they'll give you like a little page and some crayons or something like this. And in my generation, they just told us to like sit down and shut up. But we entertain you guys. So we give you like some Chili's propaganda and some colored pencils. And so you'll have like a word find or something, and it spells out like Chili's is my favorite, or ask mom for dessert, or whatever. Or like, here's a chili man, here's a chili man, here's a chili man, you know, which two are identical and all that. So at Fresco's, they gave you wiki sticks, and they did almost like a paint by numbers thing, and it'd be like, take a green wiki stick and a green one and a red one, and you like make some sort of design. So I saw these wiki sticks, and I was like, hey, these would be great for talking about splicing. So I'd go in there, and maybe it'd just be me and my wife that night or whatever, you know, date night. They'd be like, there you go. And I'd be like, can I have some wiki sticks, please? They'd be like, sure. And they'd also like bring me the little, like the little paper thing. And they'd be like, here you are, grown man. I'd be like, thanks. And so anyway, I got some wiki sticks, and the way that it works is like this. I've only got two. Hang on, I had some reds and greens in here. Give me 1 second. Or they might give it to my son, and I'd take them. He'd be like, thanks for the wiki sticks. And I'd be like, give me that, son. I got to teach some biology. All right. So we got red and blue. So here's what's strange about your RNA. Your DNA, which codes for RNA, you don't read it all. even within a gene, you don't read it all. So what will happen is this. When it is time to build this enzyme that helps me make muscle, so I damage my muscle, I need to turn this gene on, then I will make this piece of RNA. And it looks like this. There are red parts, blue parts, red parts, blue parts, red. So red, blue, red, blue, red. Before this RNA ever leaves for me to read it, I always cut out the blue parts. I never ever read those. So my RNA will loop together like this. I splice out the blue parts. I don't read that. My RNA right here will loop up, splice out the blue parts. I don't read that. And what I'm left with is the parts that actually like mean something. So this one, I don't know how well you can see that, but that's the letter R, an A, and a T. So this just has meaning. So this is the RNA that actually like leaks out that I read, and I don't read this. So it's a little bit like every time I print this cookbook, it's like, hey, here's this book. And when it's time to open it up and read the recipe, it's like, read page 1, read page 2, and then always tear out page 3, 4, and 5 and throw them away. Then go on to page 6 every single time. And you might be like, well, why don't you just never print page 3, 4, 5, and 6? Like, why do you do that and then throw it away? All right, so hang on to that thought for just a second. To conclude this, so that'll be our cliffhanger, but to conclude this, there are parts that we keep and there are parts that we don't. Whoever named this was mean. So almost always in here I emphasize that the words are logical in biology. This time they are not. So whenever I splice, I have exons and I have introns. Exon, E-X-O-N. The company has like 2 X's in it, like the oil company. But exons and introns, E-X-O-N. And then I have introns, I-N-T-R-O-N. Exons and introns. Now, using logic, you already know that it's not going to be logical, but using logic, which parts would I keep and read and which parts would I throw out? Wouldn't you logically say exons exit, so you get rid of them? We even use words like exothermic and stuff like that. Introns, you're like, I don't know, I guess they stay like inside, we kind of keep them. That's not it. Exons are the ones we keep. Introns are the parts we throw away. The person that named them said, I'll use exons because the exons are expressed. This is the genetic material that is expressed, so we'll use exons. And then logically, the ones we cut out, those pieces are intervening. So we'll call those introns, and everybody will logically get it. Oh, intron, I bet that means intervening. That is their reasoning. Exons are for expressed, which is a common way of talking about biology. So that's fair game. To the layman, it's going to confuse you, but to a biologist, it's like, okay, once I know enough, that makes sense. Introns is just a complete low blow. That's just a jerk move. So we splice out the introns, and we keep the exons. What makes splicing interesting is what we will start off with next time. So next time we'll learn how one gene can code for blue eyes, white fur, and hearing in a cat. Quiet, Trophy. See you guys later.